Biofilms formed on tooth surfaces are highly complex and structured microbial communities enmeshed in an extracellular matrix, which are exposed to constant environmental challenges. The overall goal here is to show two specific components of an analytical toolbox designed to facilitate structural and molecular studies considering the complex nature of oral biofilms by combining a novel fluorescence imaging technique with custom-made software for data processing and analysis. This is accomplished by first selecting specific assays for biofilm transcriptome and structure analyses, including a new confocal imaging approach for simultaneous visualization of EPS and bacteria. The second step of the procedure is to collect the raw data from the bioassays. The third step is to process the raw data using specific software, including the newly developed Duostat for colocalization analysis. The final step is to analyze the processed data, including the use of microarray data mining and organization software Microarray Data Visualizer, or MDV. Ultimately, results can be obtained that show how oral pathogens change their transcriptome and the architecture of the biofilms in response to complex diet-host microbial interactions using our uniquely integrated analytical toolbox, providing a more comprehensive biofilm analysis and data interpretation.
Hi, my name is Michelle Koo, an associate professor in the Center for Oral Biology here at the University of Rochester Medical Center. Today, we present an analytical toolbox for biofilm analysis. This toolbox can help answer key questions in the field of biofilm research, such as how the biofilm cells are structurally arranged with extracellular matrix and how they respond to complex environmental challenges at transcriptomic level.
My name is Xiao. I'm a postdoc fellow.